Transposable elements in eukaryotes. There are many different types of transposons in eukaryotes which vary in size, structure, composition, and behavior. All the eukaryotic transposons have basic characteristics such as having terminal inverted repeats and target specific duplication when inserted. Then types of transposons in eukaryotes. Class 1, retrotransposons. Example, TY elements in yeast, copia elements in Drosophila, line enzyme sequence in humans. Then class 2, DNA transposons. Example, P elements in Drosophila. These are cut and paste transposons similar to bacterial transposons. Retrotransposons. Class 1, donor DNA transcription RNA. Intermediate, reverse transcription DNA. Intermediate, integration, transposable element. Target DNA, transposable element. Then DNA transposon class 2, donor DNA, excision, DNA intermediate, target DNA, integration, transposable element, and target DNA. So reverse transcription happens in case of retrotransposon. Class 1, retrotransposons. Retrotransposons are those transposable elements that utilize reverse transcriptase enzyme to convert RNA intermediate into DNA and then transpose into new position by itself. They are of two types on the basis of long terminal repeats or LTR. LTR retrotransposons or retroviruses like element. All LTR retrotransposons have the same basic structure, a central coding region flanked by long terminal repeats or LTR, which are oriented in the same direction. The LTR are typically a few hundred nucleotide pairs long sequence having short inverted repeats at both ends, like present in other types of transposons. Due to the characteristic LTR sequence, which is similar to that of retrovirus, the transposons are known as LTR retrotransposons. The property of LTR retrotransposons is similar to retrovirus infection. LTR retrotransposons utilize reverse transcriptase enzyme from retrovirus that convert transposons RNA into DNA in the first stage, and in the second stage the synthesized DNA is transposed into new location in the chromosome. TY element of yeast and copia element of Drosophila. TY element in yeast. TY element is a LTR retrotransposon present in yeast which is about 5.9 kilobase per long sequence having LTRs of about 340 base pairs long at both ends. Most yeast strains have about 35 copies of TY1 element in their genome. TY1 element have only two genes, TYA and TYB, which are homologous to the GAG and POL genes of the retroviruses. Transposition of TY1 element involves reverse transcriptase enzyme. After DNA is synthesized from TY1 DNA, a reverse transcriptase encoded by the TYB gene uses it as a template to make double-stranded DNA. Then the newly synthesized DNA is transported to the nucleus and inserted in targeted site in the genome, creating a new TY1 element. So structural protein is there, reverse transcriptase enzyme, TYA gene, TYB gene, and TRLTR. The TY1 element is transcribed to produce RNA, TY1 RNA. The RNA is reverse transcribed into DNA by the enzyme encoded by the TYB gene, TY1 gene. The DNA is inserted into the chromosome, creating a new copy of TY1 element. Transposition of the yeast TY1 element. Next, non-LTR retrotransposons or retroposons. The non-LTR retrotransposons are common retrotransposons present in eukaryotes. They are simply known as retroposons or non-viral transposons. Non-LTR retrotransposons lack long terminal repeats or LTRs at the terminal end. However, they have homologous sequences of 80 base pairs at one end, which is derived from post-transcriptional modification. These non-LTR retrotransposons are transported by RNA intermediate, which is reversed transcribed into double-stranded DNA by reverse transcriptase enzyme encoded by the element itself and then transposed to the target site. Transposable element of human line long interspaced nuclear element. Sign short interspaced nuclear element. Transposable element of human. At least 44% of human genome is repetitive, which is derived from transposable elements, including retrovirus-like element 8%, retroposons 33%, and several families of elements that's transposed by a cut and paste mechanism 3%. Class of human transposons, lines, enzymes. Long interspaced nuclear elements. L1 retroposon is the principal human transposable element which belongs to long interspaced nuclear elements class. Two other line sequences, L2 and L3 line, are also present in human genome. However, both are transpositionally inactive. The complete L1 element is about 6 kb long sequence with an internal promoter that is recognized by RNA polymerase II and two open reading frames ORFs. ORF1 encodes a nucleic acid binding protein. ORF2 encodes a protein with endonuclease and reverse transcriptase activities. The human genome contains about 3000 to 5000 copies of complete L1 elements. Nucleus, L1 element, chromosomal DNA, new L1 insertion, then cytoplasm, ribosome, ORF1 polypeptide, ORF2 polypeptide. A complete L1 element inserts in the chromosome is transcribed into L1 RNA. The L1 RNA is polyadenylated in the nucleus. So transposition of L1 element involving transcription of a complete L1 element into RNA in the first step and the reverse transcription of this RNA into DNA in next step. Both transcription and reverse transcription process take place in the nucleus. However, before the L1 RNA is reverse transcribed, it travels to the cytoplasm where it is translated into polypeptides, nucleic acid binding proteins, endonucleases, and reverse transcriptase. The polypeptide remain attached with RNA when it returns to the nucleus. The polypeptide encoded by ORF2 possesses an endonuclease activity that catalyzes cleavage of one strand of the DNA duplex at targeted site in a chromosome. The poly A tail of polypeptide juxtaposed with 5' end of cleared DNA and reverse transcriptase activity results into synthesis of single DNA strand. Utilizing 3' end of a primer for DNA synthesis, the newly synthesized L1 DNA is subsequently made double-stranded. Therefore, new L1 element is placed in new location. Then short interspaced nuclear elements, signs. The short interspaced nuclear elements are the second most abundant class of transposable elements in the human genome. Signs are less than 400 base pairs long and have an internal promoter but do not encode proteins. Like all retroposons, signs do not have terminal repeats, but instead they have a sequence of 80 base pairs at one end. Transposition of sign involves transcription of sign DNA into RNA, which is then reverse transcribed into DNA. However, sign do not have any open reading frame to encode any enzyme. Therefore, it depends upon sign's type element for reverse transcriptase enzyme and other enzymes. Thus, the sign is dependent on the line to multiply and insert within the genome. Three families of signs are present in human genome: ALU, MIR, and THR2 MIR3 elements. However, only the ALU elements is transpositionally active. Then class 2, DNA transposons. Some mobile elements found in eukaryotes are DNA transposons, and the mechanism of their transposition is similar to those of bacteria. The first transposable element discovered by M.C. McClintock in maize are now known to be DNA transposons. However, the first DNA transposons to be molecularly categorized were the P elements in Drosophila. P elements present in Drosophila resemble to insertion sequence of bacteria, which possesses a short inverted terminal repeats at both ends and and a single open reading frame with encoded single protein or transpoase. The P element vary in size ranging from 0.5 to 2.9 kb in length. P elements were discovered by Margaret Kidwell, who was studying hybrid dysgenesis in Drosophila, hybrid dysgenesis in Drosophila, dystenic cross, female lab stock, male wild, M cytotype, P cytotype, F negative, defective hybrids, dystenic progeny, no offspring, but female wild, male lab stock, P cytotype, M cytotype, those in normal flies, F1. In hybrid dysgenesis, a cross between a female from the laboratory stock and a wild male yields defective progeny. A phenomenon known as dysgenesis is present in Drosophila when laboratory reared female flies were crossed with wild male flies. In such crosses, the laboratory stocks are said to possess a M cytotype or cell type, and the normal stocks are said to possess P cytotype. Laboratory flies do not have P element. In such crosses, M female, P male, the F1 progeny show a range of surprising phenotype changes in germline, including sterility, high mutation rate, and high frequency of chromosomal aberration and non-disjunction. This F hybrid progeny are biologically deficient and known as dysgenic. The phenomenon is called dysgenesis. However, in reciprocal clause between P female and M male, no dysgenesis is observed in F1 progeny. To explain the phenomenon of dysgenesis, the simple answer is that P element transposition. In all adult P cytotype flies, they possess P element which contain a transposase gene are silenced. However, the genes are activated in the F1 generation during the dysgenesis when crossed M with P. In the crosses between M cytotype female flies, no P elements, P cytotype male flies with the P element, each parent contributes respective gametes with respect to cytotype. Cytotype male gamete provides P element in the newly formed zygote, which is a silencing free environment since female gamete is M cytotype. The P element derived from the male genome can now transpose throughout the zygote genome, causing a variety of damage as they insert into genes, and all F1 progeny developed from such zygote are dysgenic. On the other hand, the reciprocal cross between P cytotype female flies P element into M cytotype male flies no P element. No dysgenesis is observed. It is because, in this case, female gamete provide P element in its newly formed zygote, but the P element is already silenced in female gamete due to presence of some component in cytoplasm of female gamete. Therefore, zygote developed from such gamete with already silenced P element prevent dysgenesis. So, insertion of P elements causing the mutations underlying hybrid dysgenesis, M cytotype, P cytotype, P generation, no P element, embryonic germ-line cell, no transposase gene silencing, mobilization by P transposase, P element, same cell a little later in time, development to adulthood, carries new insertion or mutations in germline, flies sterile, F1 generation. So that's something happening in this case. Thank you for listening.